Hello, everyone. In this video, we're going to be looking at the endomembrane system. The endomembrane system is responsible for regulating proteins, um, the protein traffic, meaning where you send proteins, and performing basic metabolic functions within the cell. So the endomembrane system consists of the nuclear envelope, the double membrane system that surrounds the nucleus, the endoplasmic reticulum, which contains both the smooth and rough ER. And then there's the Golgi apparatus, lysosomes, vacuoles, and then the plasma membrane itself. These components are either continuous or connected via transfer by vesicles. Vesicles are, I like to think of them as like little bubbles of plasma membrane that contain items that can be traveled uh, throughout the cell or even outside of the cell. So the first thing let's talk about is the endoplasmic reticulum. And this is your biosynthetic factory. The endoplasmic reticulum is responsible for more than half of the total membrane that is found in eukaryotic cells. The ER is basically several folds of the double membrane system. It's connected to the nuclear envelope, connected to the nucleus, because they go hand in hand. The nucleus is responsible for the the controls, the directions, the information that is needed to do all of the chemical reactions and protein synthesis that is needed in the cell or outside the cell. And so the endoplasmic reticulum is where a lot of these reactions occur. So there are two different regions of the ER. There's the smooth ER and then the rough ER. The smooth ER lacks ribosomes, so that's why it's called smooth. When you look at it in the underneath the microscope, the smooth ER is very smooth. It doesn't have the bumps, which are the ribosomes, which are distinct in the rough ER, where the surface is filled with ribosomes. So if you was to look at it, a diagram, for example, you can see the nuclear envelope is attached to the ER, where parts of the ER or the rough ER is filled with ribosomes, and in the smooth ER, for example, the outer rim here does not have ribosomes. Now, this is part of like an example of the endomembrane system. And so the rough ER and smooth ER make up the endoplasmic reticulum, and then they will do specific functions and create different and synthesize different things and then transmit them in vesicles. So this is an example of a vesicle that will be transmitted to different parts of the cell. If you looked at it in a TEM or on a light microscopy, you can see the rough ER is filled with the bumps with the ribosomes, and the smooth ER is smooth. It doesn't have your ribosomes. Now let's talk about the differences between smooth and rough ER. The smooth ER, um, its main job is to synthesize lipids. Remember, there's a lot of lipids that we learned in chapter 5. Not only are lipid macromolecules important for energy and the functions of the cell, but you have lipids that are responsible for building, for example, plasma membranes, the double membranes. The smooth ER is made up of a membrane. So uh, to grow or to expand or even to divide, you have to be able to synthesize lipids. Another job of the smooth ER is to detoxify drugs and poisons. They will take in drugs or poisons and they will go through a detoxification process, and it's able to clean those from the system. Uh, and so let's relate it to something that's practical. Remember, I stated that structure is directly related to function. And so if you are a cell and you have a lot of smooth ER, you're probably related to a function that is de- um, based on detoxifying. So if we think about in humans, the organ that is responsible for detoxifying things in our bodies, in our blood, and that would be our livers. So your liver cells are filled with a lot of smooth ER. They have a lot of smooth ER in the cells of the liver. And the liver cells then are responsible for detoxifying our blood um, and keeping it clean. The smoothie R is also responsible for storing calcium ions, which is important when we're talking about, for example, some chemical pathways that are in the body. Now, the rough ER is a little different in that it has ribosomes that are bound to the plasma membrane, uh, the bilayers that form the rough ER. And these ribosomes, which secrete glycoproteins, um, these are proteins that are covalently bonded to carbohydrates and which are responsible for a lot of the functions and chemical functions that are needed within the cell and outside of the cell. The roughy R will transport the products that it makes in the vesicles, and then it will send those to different parts of the cell. Generally, well, really, it will send it straight to the Golgi, which would then direct it to different parts of the cell or either outside the cell. And that takes us to the Golgi. The Golgi apparatus is the shipping and receiving center for your cells. The Golgi, I like to call it the flattened pancakes. It's flattened sacks, which we call cisternae. The Golgi is important for several different reasons. It modifies the products of the ER, the endoplasmic reticulum. So in either the smoothie R or the roughy R, when they create products that are needed to be either sent to different parts of the cell or to be sent outside of the cell, it will first be sent to the Golgi. And the Golgi is responsible for sending those products to the correct spot. The Golgi can also manufacture certain macromolecules. So sometimes you need to, you know, the Golgi is like a housing spot for ingredients. And when all the ingredients are in place, you can then assemble larger macromolecules that need to be sorted and packaged into transport vesicles that will then be sent to different parts of the cell or outside of the cell. Now, this is the diagram of the Golgi apparatus. And you can see there, I call stacks of pancakes, which are the cisternae. Now, there are two different sides to the Golgi. There's the cis-facing side, which is the receiving side, and then there's the trans side, which is the shipping side. And you can see in the diagram here, you have your trans side, which is where you'll receive vesicles, and then the shipping side is the trans side. So the Golgi will get many, many components, and it's responsible for sending it to the correct space, the correct spot. Now, sometimes things need to be recycled or digested. And so that's where lysosomes come from. Lysosomes are I call it the garbage disposal or the recycling centers found inside of your cells. A lysosome is a membranous sac of hydrolytic enzymes that can digest macromolecules. Lysosomes are very they do a really good job of what they're supposed to do. They're filled with enzymes. Um, it's a lot of enzymes that are able to um, break down many different components inside of that organelle. They work best in acidic environments. The enzymes, which are inside of the lysosomes. So sometimes lysosomes can are in, are used to either recycle materials inside of the cell, things that don't work anymore. For example, if it's an organelle that's no longer functioning, you need to break it down and recycle it. It's also used to break down things that are taken into the cell. So sometimes cells will undergo phagocytosis, where they will engulf something and then it goes into the cell and it needs to be broken down. And this is where lysosomes will do that. Lysosomes will fuse with those food vacuoles that have been created, the, these storage components, and it will break them down. And so, what you're seeing here in this image is the two different ways that lysosomes can work. If if for example, phagocytizes something, it brings something into the cell, the lysosome will fuse with that, and its digestive enzymes would then break it down into its chemical components. Same thing if something needs to be recycled, which is called autophagy, meaning that it needs to be broken down and recycled. It no longer works. The lysosome will fuse with it. In this example, a mitochondrion is no longer working. The lysosome will fuse with it and break it down to its chemical components. And all of these components can then be used again to build new things that are inside the cell. Uh, Word of warning though, lysosomes are they're really good at what they do. And so if they break open and the digestive enzymes escape the lysosome and are able to go, be exposed to the rest of the cell, they will continue to digest the cellular material. So lysosomes that break uh, accidentally kill the cell uh, that it's in. Let's talk about vacuoles. Vacuoles are these basically big storage compartments that are inside cells. Vacuoles are large vesicles. They're big bubbles of plasma membrane that usually are created from the endoplasmic reticulum and or the Golgi apparatus. Vacuoles are responsible for many functions. Um, One of the main ones being storage. So, for example, food vacuoles can be formed by phagocytosis. When you need to take something into the cell, you're going to form a food vacuole. Um, for example, there are what they call contractile vacuoles, which are found in a lot of freshwater produce like amoeba. And so these organisms live in the water, and sometimes they take in too much water. So water will be pumped into their contractile vacuoles, where they will contract and basically like spit out the water from the cell. And so one of the ways they control water intake. Plants are notorious for having central vacuoles. They have a very large central vacuole that plants will use to contain um, many different things. They can use it for food storage. They can use it for pigment storage, uh, or it can be just for nutrient storage, which is here. What the slide is showing you is a solution called sap, plant sap. Um, yes, so central vacuoles are very important because they allow for the storage of many different things. They could be pigment. You ever wonder why flowers are the different colors that they are? It's because the central vacuole is very large and it's filled with many different things, sometimes pigments, which will give plants and flowers their very pretty colors. And so in this image, you can see this is a plant cell that is got a very large central vacuole, and you can see how large the central vacuole is compared to the rest of the cell. So let's go over a review of the endomembrane system. It's a very complex and it's a very important driver for um, cells organization and synthesis of proteins and products. So this image represents the entire endomembrane system. You have your nuclear envelope where it's attached to the nucleus, and then you have your ER, your smooth and your rough ER that are responsible for synthesizing and creating different things. And it will send those products to the Golgi, which is your shipping and receiving warehouse, and the Golgi would then send those products either to different places in the cell or into outside the cell. And then, of course, if something needs to be recycled, there are lysosomes that are able to do that. In the next video, we are going to be looking at the energy processing that happens in the cell, which are the organelles, mitochondria, and chloroplasts.